Okay, so one thing that Mendel didn't know was he really didn't know what was going on with like his inheritance pieces, right? You know, he thought of the like this idea of homozygous dominant and dominant versus recessive situations, but he didn't even know what they were called. And it wasn't until like the early 1900s, which was years after Mendel, that we learned about meiosis and had an idea of what was being inherited. And of course, today we know for sure that Mendel's heritable factors. No. Hereditary factors. Or genes. And of course, genes we know are on chromosomes. And these days we're good enough that we can, if you will, highlight the location of a gene on a chromosome using fluorescent proteins. So we can use fluorescent techniques to identify exactly where one gene is located. And so with the Human Genome Project, for sure, we are not only identifying what is in your chromosomes, we're identifying where your certain genes are. So the Human Genome Project will determine the location of specific genes on chromosomes. Now, speaking of chromosomes, we know that chromosomes undergo two events in meiosis. We've talked about them before. They undergo the segregation event, and they also undergo independent assortment. Of course, that's in meiosis. And what that means, essentially, is that their movement, like which gamete gets which chromosome, is entirely by chance. Well, not really. It isn't 100% by chance. And actually, that's what this chapter talks about, is why is maybe this segregation event not by chance? Why do you have genes that are seemingly inherited together? For instance, this isn't to poke fun, this is just to be logical. Seemingly people that have freckles also have red hair a lot of times, right? That means that they must be inherited together as a combo. Okay.
Is it possible to have red hair and no freckles?
We'll actually see why that is possible, but it's unlikely. One thing that Mendel did was that he concluded that genes were unlinked. In fact, that's what I would say about Mendel, was that Mendel's pea plants had genes that were unlinked. And actually we have found, I believe, that one of his genes was actually linked up with another gene, but the linkage was actually pretty minor. So it actually still appeared like that 3-to-1 ratio that he saw. So for the most part, his genes were unlinked. Now fast forward into the late 1800s, early 1900s, and you have this guy named Thomas Hunt Morgan. And what he did is he discovered the link between Mendel's work and the discovery of meiosis. So essentially his work was all on chromosomes. And to do this, he studied fruit flies. which are gross little creatures. Literally, they are the flies of fruit. Leave out a banana for 2 weeks, there's a fruit fly. It's really gross. The reason that fruit flies actually were useful is one, because they have a high breeding rate. Do what? Right. Their generation time is like on the scale of 2 weeks or so. And also, they only have 4 pairs of chromosomes. So you could say that they have a total of 8 chromosomes. And Morgan came up with some other key terms that I think are valuable to mention. He came up with 2 terms called the wild type and the mutant type. The wild type is what we would call normal, so a normal phenotype. But the mutant type would be the abnormal or unique phenotype. Let's go over Morgan's first experiment. In his first experiment, he mated male flies.
My sister's glasses.
Oops.
My sister?
Oh, your
Oh, you
sister's.
said oops. Sorry, I thought you said news.
So he mated male flies, and they had white eyes. And he crossed them, of course, with females with red eyes. The white eyes actually are the mutant type, and the red eyes would be the normal or the wild type. The symbol for male is going to be the circle with the arrow on it. You probably have seen that before. For female, the circle with the X on it. You can make any kind of joke you feel is necessary to remember those, but you probably want to make something to remember those. Good? Fabulous. Okay, so here's what happened. He mated these 2 guys, and they all had babies. And what he saw in their babies in the F1 generation is in his F1 generation they all had red eyes. And then he took, of course, you can't self-pollinate or self-create, self-mate flies. They're not like flowers, right? Just like anything else that's an animal, you can't make its own baby. So he had to cross an F1 with another F1, a guy with a girl. But here's what he saw in his F2 generation. He saw that there was 3 red to 1 white as far as the eye goes. And you kind of see that 3-to-1 ratio. You kind of were expecting that from Mendel stuff, right? However, the ones with the white eyes was seen in only the males. Only the males had white eyes. So the determination from this experiment would be pretty simple, and that determination would be that the gene for eye color is on the X chromosome. Good. So it would make sense going back through this that the original female, which was XX, would have been big T, big T. And the original male just would have been XY, but the little t for the X. Now, all of their children had red eyes, so their children would have been, if you think about it, X big T, X little t, X big T, X little t. I'm doing the cross in my head. X big T, Y, and X big T, Y. So all of their daughters would have been carriers, but certainly would not have had it. All of these would have had the normal eyes, if you think about it, because this would be a recessive inheritance model. So all their daughters are carriers. Well, mating these two, because it's really irrelevant, they're both, all females and all males are the same. You mate those guys and you see this ratio, X big T, that could be created. X big T, Y, that could be created. X little t, let's see, X big T, and you can see X little t, Y. Okay, so that's a 3-to-1 ratio of having the white eyes. This would have had red, red, and red. Correct? See how I did that cross in my head? We good with that? Okay. But it would make sense, yeah, that 3-to-1 ratio. Except you only see the white eyes when they're a male in the XY form, not in the female. Cool? So really with this, Morgan was doing two things. One is that he was showing like the first good example of sex-linked inheritance, and I think part two of that was that he was also proving that you had these genes on chromosomes, these units called chromosomes, that fit into the meiosis thing, right? Okay, perfect. Any questions about this? One thing I want to add to this page is when I wrote fruit flies, you should write Drosophila.